Hello everyone, I'm Trong Tran. I'm a lecturer in chemistry at University of the Sunshine Coast in Queensland, Australia. I would like to thank Bridget and Kelly for inviting me to have a talk about my propolis research in this conference this year. The topic I'm talking today is antioxidant activities and chemical diversity of Australian honeybee propolis. In my presentation, I will introduce briefly about propolis and overview propolis biological properties. I will move to chemical composition of propolis, and finally I will talk about Australian propolis and my research to contribute to the development of Australian propolis industry. I'm pretty sure most of you here know propolis. Briefly, propolis is the resin materials from different parts of plants mixed with beeswax to protect beehives against pathogens and other predators. So propolis composition will contain phenolics and terpenoids coming from resins, beeswax, and other minor components such as pollen, soy, dead bees. Although Australia is lucky to have both European and native stingless bees, which can produce propolis. In my presentation today, I will mainly talk about propolis from European honeybees, Apis mellifera. On your right-hand side, the photos show you a yellow and sticky propolis produced by the European honeybees in a hive with and without propolis trap. Traditionally, propolis was used as a wound healing agent and natural antibiotics. However, modern studies have proved that propolis has a wide spectrum of biological activities as listed in this slide. More recently, one study in Brazil and US found Brazilian green propolis can facilitate SARS-CoV-2 treatment. Please notice that not all propolis has these properties, as they depend on the chemical compositions of the plant's resins, bee forage. Good quality propolis should have more resins having bioactive compounds and less beeswax. As a consumer, we often see propolis is used as an active ingredient in dietary supplements, medicated products, cosmeceuticals, and food. On your right-hand side, there are some propolis products that you can find in our chemist or pharmacy store in Australia. In 2020, I published a paper discussing about the chemical compositions of both honeybee propolis and stingless bee propolis. The studies collected chemical structures of 578 compounds, which was isolated from honeybees and stingless bees propolis and characterized by NMR or X-ray crystallography method. But I only want to focus on honeybee propolis chemical compositions in this presentation today. As you see in the chart, Americans reported the most compounds identified from honeybee propolis, while Oceania, including Australia, reported propolis compounds very limiting. From 502 compounds identified from honeybee propolis, about 80% of them are phenolic compounds, which are classified into nearly 30 phenolic subclasses, with a statistic as shown in this slide. Terpenoids occupies about 90%, with mainly diterpenoids and triterpenoids. So far, no alkaloids were isolated from honeybee propolis. Before I move to Australian propolis, I think I should mention four well-known and well-studied propolis types in the world. They are poplar propolis, Brazilian green and red propolis, and Pacific propolis. Poplar propolis is only found in temperate countries such as US, Europe, China, New Zealand, and South America. Red and green propolis are in tropical regions in Brazil. Pacific propolis is in the subtropicals and tropical regions and can be found from Okinawa, South Japan, Taiwan to some Pacific islands. Why they are considered as hydrate propolis? Because they contain a high level of phenolic compounds, high recovery yield, and so strong biological activities such as antioxidant, antimicrobial, anti-inflammatory, anti-cancer. So when you look at these figures, you will find the resins of these propolis come from different parts of the plants. Poplar propolis come from bud resins, Pacific propolis from young fruit resins, Brazilian green propolis from leaf resins, and Brazilian red propolis from bark resins. How about Australian propolis? Australian propolis is still a mystery. Very little information about chemistry, biological properties, and also botanical source of Australian propolis so far. As you might know, Australia is ranked as the seventh most biodiverse country in the world, having over 6% of world plants with 86% of native plants. So we should expect Australia provide diverse and unique propolis type, which cannot be found in any regions in the world. In fact, only propolis in Kangaroo Islands, South Australia, has been fully studied so far. Its product is currently being commercialized, as shown in this slide. Kangaroo Island propolis contained high level of phenolic compounds, with some unique stilbenes having more potent antioxidant than the Brazilian green propolis and more potent anti-cancer activities than the anti-cancer tamoxifen. It was found that sockset rats and kangaroo thorn plants are two sources providing resins for Kangaroo Island propolis. My lab at University of the Sunshine Coast is running a screening to identify propolis with high level of polyphenolic compounds for further development. As we all know, polyphenolic compounds have strong antioxidant activities, and this is a key factor to boost human immune system and reduce the risk to have other chronic diseases such as cancer, infection, inflammation, heart, diabetes, and neurological disorder. Targeting high polyphenolic propolis will help us find premium products for commercialization and potentially discover special bioactive compounds for the development of nutraceuticals and pharmaceutical products. This flowchart shows you how we are doing in the lab. Raw propolis is ground homogeneously and then extracted in 70% alcohol to remove wax. Alcohol is then removed under vacuum to obtain pure propolis resin. Following assays including total phenolic contents, total flavonoid contents, and antioxidant activities are conducted to assess the qualities, particularly antioxidant activities of propolis. My lab is also doing chemical profiling using LCMS and NMR. This database will be valuable for future study about authentication and traceability of Australian propolis. We completed a pilot study for Australian honeybee propolis in 2 years. We obtained over 200 samples which were donated by beekeepers across the country. A map on your right-hand side reveals the number of samples and their geography where the propolis was collected. Although these samples absolutely did not cover the whole country, this pilot study will give us a snapshot of the quality of Australian propolis and if Australian propolis is diverse and unique as our hypothesis in the previous slide. On figures at the bottom of this slide give you a small subset of propolis samples in the studies with different color brown, dark brown, black, red, red-brown, orange, or yellow. They have intense aromatic resins and sweet honey flavor also. These graphs show you the results we found from the pilot study. On the blue dots are Australian propolis samples. The orange dot is poplar propolis from New Zealand and Uruguay. Green and red dots are Brazilian green and red propolis. I included poplar and Brazilian propolis in my study as controls to assess the quality of the Australian propolis. On propolis extracts was tested at 100 microgram per ml to scavenge 100 micromole of DBBH free radicals. Australian propolis scavenged 59% of DBBH on average, while the reference samples shows over 90%. However, we also could find some Australian propolis show antioxidant activity comparable with the Brinkmann propolis as reference. The total phenolic contents of Australian propolis vary from 1 to 180 mg gallic acid equivalent per grams of propolis extract, with an average of 68 gram gallic acid equivalent. Total flavonoid contents of Australian propolis ranged from 1 to 114.5 mg quercetin equivalent per grams of propolis extract, with an average of 24 mg quercetin equivalent. Similarly to the antioxidant activities, results of total phenolic contents and total flavonoid contents show us there are some Australian propolis types with comparable or even better activities compared to popular and Brazilian green or red propolis. Because the study was conducted after the droughts and bushfires happening in Australia in 2019 and 2020, we think the drought and bushfires affected negatively to the findings and worsened the results obtained. To investigate chemical diversity of Australian propolis, I separated my propolis database into two groups based on their total phenolic contents. Group 1 is high-rate propolis, with total phenolic contents is over 75 milligrams gallic acid equivalent. Group 2 is low-rate propolis, with total phenolic contents is less than 75 milligrams gallic acid equivalent. The untargeted multivariate statistical principal components analysis based on their proton NMR signals indicated there are significant discrimination in chemical composition of the high-rate propolis collected in Queensland, South Australia, and Western Australia, where Tasmania, New South Wales, and Victoria propolis don't show any significant difference. The low-rate propolis group don't show any significant discrimination in chemical compositions among samples collected across the country. The chemical profiling study on the current database revealed Australia has 16 high-rate propolis types, and generally they are very unique in each Australian state. Queensland has 4 high-rate propolis types, South Australia has 5 unique types, Western Australia has 3, New South Wales has 2, Tasmania has 1. And there is one propolis type which dominated samples collected in New South Wales, Tasmania, and Victoria. Interestingly, their chemical profile is similar to the popular propolis in Uruguay and New Zealand. So you can find results of my study in details in the scientific report paper, which was published a few months ago. In addition to screening the antioxidant activities and chemical profiling, my lab is also running research to identify bioactive compounds in propolis as described in this flowchart. We are trying to discover compounds with potential for the applications of nutraceuticals, cosmeceuticals, and pharmaceutical products, such as antimicrobial, anti-inflammatory, anti-cancer, anti-diabetics, and wound healing compounds. This is an example of what we found and published. We did a screening of over 200 propolis extracts and found 5 propolis extracts showing potent inhibition against alpha-leukosidase and comparable to the anti-diabetic drugs acarbose, which was also included in the screening as the control. 3 out of 5 propolis extracts were from Queensland and had similar chemical profile, which belonged to one propolis type. After multiple steps of bioassays and chromatography, 6 active breath-related flavonoids was isolated. A full result of this research was published recently on food journals as shown in this slide. Interestingly, chemical profile of the bioactive Queensland propolis is relatively similar to the profile of Pacific propolis in Taiwan, reported previously. The only difference is Queensland propolis has one flavonone, solophenol D, which hasn't been reported in Taiwanese propolis. Taiwanese propolis is also show anti-diabetics on an in vivo study as reported in Nutrients journals a few years ago. And currently, this Taiwanese propolis is commercialized as a supplement in Taiwan. Because Australian propolis has been underexplored, my study provided better understanding this new resource. We can see Australian propolis has sometimes with potent antioxidant activity suitable for the development of nutraceutical, cosmeceutical, and pharmaceutical products. However, they are very diverse in chemical compositions, so understanding their compositions is the next step we should do. Before my study, there were some expectations about the presence of the poplar and Pacific propolis in Australia. However, no scientific data proved that hypothesis. So my study confirmed they are both present in Australia now. With the with a current propolis library contain over 300 Australian propolis extract, I hope to find more collaborators who are interested in the discovery of new therapeutic properties for Australian propolis to work on this valuable resource and bring more values to Australian beekeepers and apiary industry. To end up my talk, I would like to thank my collaborators, Australian beekeepers, and I also acknowledged Hive and Gwena Australia, Refuge Australia, and UniSAs for the funding support. If you are interested in Australian propolis and want to collaborate with me in any research or translational projects regarding to the development of Australian propolis for nutraceutical, cosmeceutical, and pharmaceuticals, please contact me via this address. Thank you very much for your listening to this presentation.